All right, so as promised, we are live with some CSEC biology. I have dragged out, willingly, willingly uh, asked Ms. Achille Joseph to accompany us with the CSEC Bio. It's been a while since I've taken a dive into CSEC Bio, so I don't want to be making any mistakes to the nation's children. So I called one of our experts at the Student Hub, Ms. Achille Joseph, and she will help us with the CSEC Bio. So we starting, what's the time? It's 10:30. We were supposed to start at 10, but earlier we were just fighting up behind the scenes to get this thing to work. And we figure out something. So we ready to perform. So you does teach bio bread. If you look back in the channel, you'll see I used to put on some work in the bio, Actually had a little thing in the bio. But
so then you will lead us with the first question.
I wouldn't take that ladies first, man. And Mother's Day coming up, I wouldn't take that away from you.
So, okay, all right.
So, all right, how much of us we have here? We have like 50 of us here. So let's just jump into it. You can just pop up the slide there. CSEC Biology Inheritance Live. So why are we doing inheritance?
Well, so usually when we get to that section C, you start off with your cell division, your mitosis, your meiosis, which you can kind of learn on your own. You learn off the steps, the stages, you know what to happen, right? And then based on that, they move into this sort of patterns of inheritance where, they start to talk about things like genotype and phenotype, like genes and genetics. And that is where students kind of get confused, as in it's a practical question. It's not straight like study for some definitions.
It's have a thing with you seen this. It have a thing with a box like this. What is it called again?
Right. So so normally when they ask for genetic diagram at this level, that's the Punnett square that you want to jump out with.
The Punnett square.
Right. You line up that's the evidence to back up any statements that you're making.
Okay.
So you have a little deduction to do. I like it because it's kind of like problem solving mystery. Sometimes you have to figure out if this person could be that person's dad based on the genotype.
Like question I got today?
Not so bacchanalish. The bacchanal is more in HSB. They like more thing in HSB like that. But it's the same concepts coming through.
All right, so let me jump into slide 2. And I guess you would take it away first there, Kelly. I would just pop in any questions if I have any questions.
All right. So I pulled the different parts of various papers that had any inheritance-based questions, right? And so we can go through it and just see, how you would line things up to give your answer, right? Mm-hmm. So this is from so on each slide, you will kind of see a box with the year and the part of the question that it came from. So in 2023 in January, they were asking about Mary. So they say attached ear lobes is a recessive trait and it is not sex-linked. Mary has attached ear lobes like one of her parents. Her other parent has free ear lobes. Using a genetic diagram, show the cross between Mary's parents. So the thing here is that you have to have a little foundation on understanding what words mean. So the fact that they mention recessive trait here, and they say that it's not sex-linked, that is supposed to mean something to you, right? So from your theory, you would know that you have, if they say recessive trait as opposed to what? It's recessive as opposed to being dominant. Dominant traits mask, and they show, for a kind of inaccurate way to think about it is like it's strong. So once it's present, it's going to make its presence known and felt. Whereas recessive traits are more laid back, more introvert, they only show their true nature when the dominant trait is not around, when the dominant allele is not around, right? So they're easily bullied. Yeah? She is speaking too fast. All right. I'll slow down, OJ.
Nah, you're speaking good speed. Don't worry, OJ.
Well, normally that's something people tell me now, so I thought I went in, right? So with that, we see that attached earlobes is that recessive trait. It's only going to be seen when the alleles for normal earlobes or free earlobes aren't around. And then they say that it's not sex-linked. So we know that we have 23 pairs of chromosomes, and 22 of those 23 pairs are called autosomes. It means that those pairs are the same for male and female. So therefore, any alleles, which are forms of a gene that is found on those chromosomes, it doesn't matter whether you're male or female, the inheritance of it has nothing to do with sex. So it's saying that the gene for air lobes is going to be on one of those autosomes. It doesn't matter whether you're male or female, so we don't have to think about whether it's on the X chromosome or the Y chromosome.
So if it was sex linked, when you're drawing the little genetic diagram, you have to like something with XY or something.
Yes. Okay. So I have some questions like that for sex linked conditions as well.
Okay. So you'll see what will happen if it's sex linked. All right. Let me jump in.
Yeah. So when they say Mary has attached ear lobes like one of her parents, it means that there is no dominant allele for free ear lobes in her genetics, right? And it means that one of her parents has the same thing too. And whether it's the mom or dad, doesn't matter, because they said that it's not sex-linked, right? So when they say using a genetic diagram, show the cross between Mary's parents, it means that we can see that mom, you have mom and you have dad, right? And we can pick mom to be the parent with free ear lobes, right? So actually, no, let's go with mom having the attached ear lobes, right? One thing that they want to do at the side in these questions is when they don't tell you how to score or code the alleles, you have to tell them what you do, right?
So the assumption.
So that so that they're not assuming. Okay. Would the examiner probably assume if you don't do it? Maybe, but just proper form, you define your alleles, right? So somewhere to the side, you will say, let big E code for the small L.
Okay.
You will let for the attached ear lobes. Just a note there, whenever you're picking letters, try and pick letters like E and G, like E, G, F, that have very distinct capital and common letters. When you use things like C and X, you know, depending on how you like, you know, it's like if you do C, or you're writing fast and you do that, they might not know whether it's capital C, or capital C, common C. So you want to avoid those that the capital and common look pretty similar, right? So if mom has attached ear lobes, just like Mary, it means that no dominant allele is around. And we use the capital letter to represent the dominant allele and the common letter to represent the recessive allele. So mommy will have to be small e, and which means Mary is also small e, because she has attached ear lobes. If dad is the other parent that has normal ear lobes, it means that he has to have a dominant allele, right? But for him to be able to have a daughter that is Mary, he has to be able to give an allele that codes for the recessive trait as well. So that means daddy, daddy cannot be big E. Yeah. He has to be big E, small e, so that he can give a small e to Mary. Right? So the genetic diagram of choice for you is to jump in with your Punnett square. And what a Punnett square looks at is looking at the possible gamete types that each parent can produce, and then showing you all the possible combinations of those possible gamete types, right? So in terms of the gametes that mommy can make, she's small e, right? And you know that gametes, so we look, humans are diploid organisms. We have 2 copies of every gene. And when you're forming a gamete, gametes have half the number of chromosomes in it. So only one of the 2 copies will go to each gamete, right? But because mommy has 2 small e gametes, it means that mommy is homozygous, 2 of the same form of the gene. She has 2. So all her gametes will contain small e. Dad, on the other hand, because he's heterozygous, he has 2 different alleles at that gene locus. He can make big E gametes and he can make small E gametes. So daddy can make two different types of gametes, but mommy can only make one type of gamete. So when you jump into your Punnett square now, so it's really a 3x3 block, right?
That's
The
the best
3x3
3
block
by
there.
3
You're going to
block
put
I've
the
ever
possible gamete types for one parent. Pardon?
No, I just said that the best 3 by 3 block I've ever seen.
Oh,
You hear me?
I'm not too sure. I'm not too sure if you're being straight or it's a kind of underhanded insult of my
I am not
diagram.
being straight. That is a horrible 3 by 3 block. But go ahead.
The judgment, right? So along the top, you're going to put your possible gamete types from dad. So you put, dad could give you half of his gametes going to be big E gametes. The other half of his gametes are going to be small E gametes. But mommy, she could only give you one type of gamete. So it's going to be small E. So then you're filling in each block now. I will call it 1, 2, 3, and 4. That's showing you the 4 possible combinations that can result from these 2 types of gametes coming together, right? So these are the 4 possibilities that can occur with every single fertilization event, right?
Britannia is saying, don't they normally use X and Y? But is she talking about sex linked?
Yeah, so we only factor in X and Y if the alleles are on sex chromosomes, but they specifically said in the question that this is not, right? So in the first box, you would have the big E gamete from daddy with the small E from mommy. In the second box, you have small E from daddy, small E from mommy. Third box will be big E from daddy and small E from mommy. And the last box will be small E from daddy, small E from mommy, right?
Okay.
So these are the 4 possible combinations that can be produced when this particular mom with this genotype and this particular dad with this genotype reproduce.
So these here, we call these the genotypes up top. These are the genotypes.
Yes.
What do you call that?
Yeah.
All right. So the next part, when they come to state the genotypes, we'll just write EE for her like that and collect marks.
So Mary has to be small e because she has a recessive trait like mommy, right?
Okay.
So you're showing the cross. So this Punnett square shows the possible outcomes from a fertilization event between this mom and this dad. Then they said, based on the results on the cross in D part 1, state the probability of a child having attached earlobes, right? So these 2 possibilities here, lead to normal lobes. And these 2 lead to attached. You're saying something?
No. I just repeated what you're saying. So 50%.
Right. So, but before we get to 50%, just to say how we get 50%, these are the 4 possibilities, right? So 2 out of the 4 possibilities allow you to be normal, and 2 out of the 4 possibilities will allow you for the attached earlobes, right? So that means you have a 50% chance of having normal earlobes and a 50% chance of having the attached earlobe. So answer based on the results, state the probability of a child having attached earlobes is going to be 50%.
All right, next slide.
Right, so this is from 2022. The copy of the 2022 paper had a terrible type over the question, right? So Ellis becomes worried that his child may also develop sickle cell anemia. His doctor tested his wife's blood and told Ellis that it is unlikely that he will have a child with sickle cell anemia. With the use of a genetic diagram, explain why it is not possible for Ellis to have a child with sickle cell anemia, right? And now they told us in the question, in your answer, let big S denote the allele for sickle cell trait and big A the allele for normal trait, right?
Hold on. Can I do this one? And then you will let me know if I get the marks.
Sure.
All right. So phenotype. Oh, geez, wait. I'm walking in unequipped here. What is phenotype?
So phenotype is the external manifestation or how the father presents. So we're talking about sickle cell anemia here. So the phenotype is going to be based on whether the person has sickle cell anemia or not.
Right. So I know the phenotype for the father is that he has it, but I want to know, like, what do I write? Do I write S? Just write S?
So I'll write sickle cell anemia because the S coding is when you start talking about the alleles.
Okay. So sickle cell anemia. Right? And the genotype, okay, phenotype of the mother is, what do we write? Not sickle cell anemia?
Right. So the fact that they said that because the doctor said that it is unlikely that he will have a child with sickle cell anemia, so it means that
the mother doesn't have anything.
That means mommy.
Yes.
So if it's not possible, that means mommy doesn't have anything.
Right. So not even the trait, right? So, but what do I write? No sickle cell anemia, no trait, no nothing. How do I write that?
You can just put normal. So they coded it as sickle cell and normal. So you
normal. All right. So the genotype of the father would be SS. It could be SS or no, SA would be a trait. So SS and the phenotype of the mother would be what they're doing for normal was the thing where that letter is over the A.
Capital E.
A. You just put AA like that?
Yeah.
Okay. And the genetic cross would be S. Well, wait, I jump in the gun. I suppose to put. I'm feeling like I'm under pressure.
I waited to see I waited to see upon it square.
And then I suppose.
So you already listed genotype already.
Okay, so I can just put gametes, right? That?
Yeah.
So I see you do this, and I see you do this. Well, if it's the same thing, I could just write it once.
You can press just for completeness. So this is the same strategy all around, but
okay,
it's different.
SS A. I know you're waiting to judge my 3 by 3 square here. Oh yeah, sorry, that was a mistake. So this is going to be, who do you put first? Let's put AS. With all of them going and come out with traits. And the A is dominant. And S is recessive. So the phenotypes of all possible offspring. I like how you say in a what's the phrase you use in a reproduction event? That's an interesting
event.
And in a fertilization event, all possible offspring in a fertilization event, fertilization event will have will be normal trait.
Okay. And you could put a note here, do that all of their offspring will be carriers.
Oh, all of their offspring will be carriers. I have this right. My genotype is AS. I am a carrier right now. No, like I mean in real life.
Right. So,
Yeah.
okay. Well, I figured that from doing, but
Yeah.
it would make sense, right?
Yeah.
Um, the so very good response. The only thing I would the only thing I would kind of comment, like to say to emphasize here, is that you have to explain why it is not possible for Ellis to have a child with sickle cell
Oh,
anemia,
okay.
right? And the reason why, so the very clear explanation for that is because mommy does not, if it's impossible, it is not possible at all, it means that mommy does not have a recessive allele to give. So her dominant allele will mask Ellis's recessive allele.
I'm
The
not too sure I understand what you're
impossibility
saying.
comes from mummies. The impossibility of Ellis having a child with sickle cell is guaranteed because mommy is homozygous dominant. Her two
Right.
alleles are the dominant allele. So every child that they have, she will give a dominant allele to. So that child will always have a normal copy, always have a dominant copy to mask the always recessive allele that Ellis will have. So Amelia is saying this question is weird. Isn't
That's
the
a very good
allele
question.
supposed to be of the same letter they give it as
Amelia,
their
question.
So that is true, right? They do weird things at times, right? But when they give you a code, you have to work with it. You can't put an learn your tricks. You just work with what it is, right?
All right. I feel I could get a mark to find if I have to do it in the
Yeah,
exam
but
now.
real good.
Yeah.
Yeah.
Number 4. Slide 4.
So the genetic diagram, your Punnett square, is just really proving that you're not seeing any sort of um, smallest combination, right?
Okay.
Now we get into blood types. This is another one that they really commonly test, right? So you have the ABO blood grouping system is determined by antigens on the surface of red blood cells. An individual who has one parent with the heterozygous genotype for blood group A and the other parent with the heterozygous genotype for blood group B may have a blood group phenotype that is different from either parent. With the aid of a genetic diagram, Punnett square, explain how an individual may have a blood type that is different from either parent, right? So again, these are where certain terms in the question have importance, right? So it is possible for two parents with particular blood types to have a child that has a very different blood type from either of them. And it's not that, and not to show the environment, it's not that infidelity occurred, right? But it is a genetic possibility, right? So the first thing that we're going to look at here is that they say that one parent is heterozygous for the genotype of being blood group A, right? Heterozygous. So to be blood type A, so this is side notes, right? This you don't have to include in the question, right? So we call this side note over here. To be blood type A, you could be AA. As in the two alleles you have, you're AA, or you could be AO, right? Sometimes you can code, so the proper way to code this, because they're talking about antigens, is to actually put it on the base of I. But if they didn't ask you to do it, I wouldn't ask that complication of you, right? We'll keep it simple, right? So that you can have two dominant alleles, AA, or you can have one dominant allele A and a recessive allele O. That's the two ways to be type A.
Right.
Then the other two, so parent one. So let's just, this is not sex linked, so we could pick either one to be mom and dad, right? So mom and dad, their phenotype is going to be type A, and that phenotype is type B. To be type B and B, two dominant Bs, or you can have a D and a recessive O. So again, in both cases, they told us that it could be heterozygous or dominant. They're heterozygous, right? In each type, it could be homozygous dominant or heterozygous. But the only heterozygous option is AO and BO. Heterozygous meaning that there are two different types of alleles at that particular gene locus, right?
Yeah,
So it means that mummies.
If you go back to slide 3, or the one that you did, slide 2, is there like something that we need to do with labeling, like on the, you see what I'm talking about? Are we supposed to put mom or dad on the Punnett square anyway, or that is understood?
Well, it is understood from the codings that we brought down, but you can label this as the options for dad, or you can even use the genetic symbol, and this would be mom over here.
Okay. All right. I already feel I know I'm going with this ABO one, but you can take it away. AO is a bounce.
So, right. So I thought it was going on in here now. So mom genotype, she has to be heterozygous, so she is definitely AO. Dad heterozygous, so he is going to be BO. Right? Possible gamete types from them. You're going to have A, half gametes being A, half being O, half being B, half being O. And we go to the Punnett square now. Right? So you can put dad possibilities here, mom possibilities here. So you can have B, O. So the 4, I like to just number it so you can follow in terms of reference, right? So one possibility is that you get the A allele from mom and the B allele from dad, and you'll be blood type AB. Or you can get the O allele from dad and the A allele from mom, but you always write the dominant alleles first. That's this common practice, right? So if B or A is present, it's always going to be written before O, right? But A and B are co-dominant. They have equal strength. They both show in the phenotype. So, but you just, oh gosh, in the alphabet, A becomes before B, so you'll write AB and not BA, right? But technically, either way is right. A is not dominant to B, right?
Okay.
So yes, this combination here would be AO. Then in box 3, you would have B allele from dad and O allele from mom. And in box 4, you would have O alleles from both parents, right? So these two possibilities here give you, I'll call it parental phenotypes. Right? But the other two possibilities here give you your box 1 and box 4 show you that there is a 50% chance of these two parents having a child that will have a blood type that is different from either of theirs.
Vibes.
Yeah?
And you could
so
probably
you
just
might
write
just
in
want to conclude with a statement saying that, or you can say, there's a 50% that can have a child, a blood type is different from, and that blood type being, they can have a child as AB and also as blood type O.
Okay. I don't know if it's your end or mine, but once or twice again, I'll stick, but it's okay. It's not bad. Okay. Somebody was asking if it was homozygous, it would be AA and BB.
Yes. So if they were homozygous, it would be these two genotypes here, but then that means all the offspring would have simply been type AB, and it's a wrap.
All right. Next question.
Yep. So this question I put in just to show. So if you want to jump in, Kerwin, on this one, it's basically the same question, but in a different, more structured format. Right?
Okay.
So a couple with blood groups A and B have a son with blood group O. Complete the genetic diagram to show how this is possible. So it's the same question, but just asking a different way, and you have a more structured approach. So somebody has to go over it. We're going over it now with discussion from 2019, right? So parents' phenotype, you give your blood group A and blood group B. So the parents' genotype are going to be what? You know, in order for them to have a child as blood group O, they're going to have to be heterozygous.
Yeah, heterozygous AO. But wait, I'm watching this Punnett square here. Why this looking so?
It's disproportional, but the boxes are the same. It's the same 3x3, but ideally it should all be the same size.
Okay, I was just getting a little worried. So, right, so we parallel AO here, parallel BO there, parallel AO, BO, something like that. Well, it's the same thing. It's the same thing. So.
Right, you put A, B, O on one side, A, O on the other side. You show your possibilities.
Put the dominant first. And let's now get the proper terms there. These are the parental phenotypes. Phenotypes, 50% chance, total for. And non-parental phenotypes.
But so with to jump to parental, because I didn't ask about non-parental.
Okay, right. They asked for blood group O. So
yeah. So and then in part 2, they say, well, what is the genotype of the child group O? But A and B are co-dominant, but they're both dominant. To be O is the absence of a dominant, absence of A, and the absence of B. So the only way to be blood group O is to be OO. And there's only a 25% chance of that happening.
Okay. What is the... Oh, there was accident dispatch.
So hope that clears it up for Candy.
Ooh, I just deleted the whole slide. All right, yeah. Got it back.
But I was saying the image just disappeared.
Oh, yes. Yeah, when I went to Bigo, it have... I stay by somebody named OO. And her sister name is AA. I kid you not. OO and AA. Anyhow, next one.
Keep it simple. Right? So this one is where we're looking at a condition that is sex-linked. So you're seeing the X's in the mix. So because it's sex-linked, we have to consider, the sex of the individual. So sometimes persons may inherit a condition called hemophilia, in which the blood does not clot normally. The allele for blood clotting is located on the X chromosome. So they didn't use the word sex-linked here, but the fact that they told us that this allele is on the X chromosome, we know that it's sex-linked, right? A female with normal blood clotting ability mates with a male with normal blood clotting ability to produce offspring. So mom is normal, dad is normal, right? Complete the following genetics diagrams to illustrate this information. So the phenotypes, they're going to be male and female. Now, yes, male and female are the phenotypes, but that's not really the topic here. It's supposed to be a male with normal clotting and a female with normal clotting. That is the full description of the phenotype. But again, we're not grading CSEC, them grading us. So moving on. Then we have the parents' genotypes now. So the female, they're telling us that the female is X, big H, X small h, right? So they didn't really code it for us, but basically when they did that, they're telling us let X big H be the allele for normal blood clotting and X small h be the allele for abnormal blood clotting or hemophilia.
But every now and then that I still away here in the background.
Yeah, boy. I don't know. I'm really trying to get her to go, but she really
She taking in some bio. That's okay.
she's taking in some bio. So that is by them just putting that there. That's what we're going to understand here. Now we have to fill in the genotype for the male. So to be a male, what is the basic genotype to be a male?
XY.
You have to be what? You had to be XY, right? So that going in there.
Like this.
Okay.
Russia.
All right, you write it, right? So if it is that you have to be, if it's a male, you have to be XY, right? It's the Y chromosome or genes on the Y chromosome that determines maleness. So your default sex is female, and it's the presence of the Y chromosome or the products of genes on the Y chromosome that make you male, right? So that the male has to be XY. But we have to specify whether that X is going to be an X with a big H or an X with a small h. But because they told us that the male has normal blood clotting ability, it means that, do you think he has to have an X big H or an X small h?
Big.
He's going to have an X big H, right? So then the possible gametes that daddy could make would be,
and you don't put anything on the Y?
No, because remember this allele is on the X chromosome, so you're only going to talk about nuances of the X.
Okay.
Right? So then you put in any possible gametes that daddy could, and you put in your little brackets there, right? And then you fill that in the in the table, in the Punnett square.
So wait, to be hemophiliac, if you have a situation where like this person is what do you call it, hemophilia, has hemophilia.
Yeah, that would be a male with hemophilia.
Okay.
Pardon?
So this is going to play out some weird probabilities if, yeah.
So that is a particular point about sex-linked conditions, that sex-linked conditions disproportionately affect males more than females. And the reason for that is that males only have one X because they have a Y. Females have two X's, so the chances of us having a good or a normal X to back up and mask to be dominant to that particular allele is very high. So we generally have a backup while males don't. So if they get have the condition, female bad copy, but have a good backup to mask it.
Way. So he and have problems there.
Right? So this question has a part 2 and a part 3, so we could go on to the next slide.
All right.
So wait, before we move on, so just to highlight here, we have your possibilities from mom. Mom has a normal for that hemophiliac or abnormal clotting allele, right? But because she has a normal copy, she has a backup. She's phenotypically normal, and they described her as being normal blood clotting, right? Dad is normal as well. He only has one X, so he has to have a good copy of X. And then obviously because he's male, he would have the next one being a Y, right? But this is showing you here that there is a 25% chance that they can have a child with hemophilia, right? Out of the 1, 2, 3, 4 possibilities, this is the only possibility that he can have a child. So it is one, impossible for them to have a daughter, that is hemophiliac, and one that is a phenotype that is, that is a genotype that is not compatible with life. So generally, if there's a female with two alleles, so X small h, that genotype is not compatible with life, and those fetuses self-abort very, very early in pregnancy.
What?
So, which is not the case for all. It's not the case for all sex-linked conditions, but it is the case for this one. When we go on to do color blindness, then you will see the normal plays going out, right? So the part 2 here is that state the probability of the couple having a hemophiliac child. That probability is going to be your 1 in 4 or your 25%, right? What is the likely sex of the hemophiliac child? Give one reason to support your answer.
I don't know how I could pull out that 3 marks out of that, because I feel like that's one sentence.
Okay, so first, your 1 mark is going to be what is the sex of the hemophiliac child? That's easy.
Male.
It's going to be male. One reason to support your answer is that the mother is the one that is the carrier for this hemophiliac allele, right?
Mm-hmm.
So it's only the mother that is going to be able to pass on that allele to her son for him to have the condition. Who have X big H,
when I know what's going on.
All of his daughter X big H.
When I write in is when your mic's sticking, so
right?
You are right.
All right. The reason so is the impossibility here is that dad or his daughters and big H allele, right? So it would be impossible for daughter. If it could have persisted, it's impossible for a daughter to have the condition. Follow?
All right. Yeah. And that's it. Next line.
Yep.
Well, scroll back up. Well, she could always watch over. Pull back. Leave it.
So color blindness is another condition that is sex-linked. A colorblind woman mates with a man that has normal color vision. Determine the probability of this couple having a colorblind daughter by completing the genetic diagram below. So the parents, phenotype, normal color vision man, colorblind woman. The parents' genotype is going to be X big CY, and the mother is X small c, right? So mommy could only make one type of gamete. So she is given this colorblind allele to all her offspring, right? Normal vision man, well, he's going to be giving Y's to his sons, and he'll be giving a normal X to his daughters.
Yeah, I don't really watch it with these big C and small C, boy.
Yeah, so the C is a tricky one. That C is one of them tricky letters. So you had to be clear. Real exaggerated.
Way, boy, them fellas have it real rough out here, boy. What's going on here?
Right?
But anyhow,
so,
probability of a colorblind.
Following what we, that is what it is. So that is what you won't believe.
That's okay, Stella. Just
and there's nothing. bothering her, you know. Anyway, so the probability of the so what is the probability of having a colorblind daughter here?
0.
0 is impossible, right? So it is impossible for them to have, because daddy is going to give that normal to all of his daughters.
You're cooking with Ramco gas.
Yep.
Oh, by the way, when they say fertilization cross, they're referring to this, right? So I don't need to write anything here, right?
Yeah.
Okay.
So these are the possibilities of a single fertilization event. And the reason why we kind of emphasize that is because some people kind of misinterpret this diagram to mean that these are the 4 children that these 2 people have. That one child is X big C, X small c, one child is no, but when it is time for them to have this one child, these are the 4 possibilities that that one child could have been, and then you figure out what the actual status is.
Okay. Next slide.
Right? So this is the next one where they code and tell you a little madness, right? So a certain form of color blindness is inherited as a recessive allele carried on the X chromosome. It is thus said to be X-linked or sex-linked. Normal color vision, whose father is colorblind, mates with a colorblind man. What is the chance of them having colorblind children? Use a genetics diagram to explain that. And the madams here now, where they're not telling you to factor in so they're telling you this is how they code it. Use big capital B for the allele for normal vision and small b for the allele for color blindness, right? Technically, that should be X big B and X small b. Yeah?
So I would go ahead and do that.
So the easiest thing for you to do is to go ahead with that, because when you don't factor in the X and Y, things is gonna get a little muddy real quick.
All right, right.
Yeah. Right? So right? Mom and dad. Off the bat, mates with a colorblind man, so we know that dad is
small b.
X small b. Sorry. All right, now we still on phenotype. Dad is colorblind, and a woman, mommy is normal, right? So dad's genotype is going to be Y, right? Mom is normal. So that means mom could be X big B, or X big B, X small b, right?
But wait, her father is colorblind, so that means she has
2 possibilities for mom, right? Right. So how we know which one mommy is, or how we narrow it down, is because mommy's dad is colorblind, which means that he would have given her the X with the small b.
Got it.
Understood? So that means mommy genotype, she can't be homozygous, heterozygous. She has X big B, X small b.
And then you organize it.
Right? So moving on with that. So after we know for sure what the genotypes of the parents are, use a genetics diagram to explain what is the chance of them having colorblind children. So then you have your gametes. Right? You go on with your Punnett square. You show your possibilities 1, 2, 3, 4, but again, just for reference as we talk. So in the first block, you're gonna have your X big B. So this is mom on this side and dad on this. I have a very sexist way of remembering which symbol is which. I ain't too sure if I should share that one now. But because students, sometimes they don't always put male and female next to the symbols, and sometimes you have to know what it means, right?
How do you remember? Say it, right? No. My YouTube channel could take these kind of things. My viewers are well cultured.
Yeah, sure. Well, it's not the viewers. I mean, I consider body viewers, but I also interested in maintaining my employment. I know I like to pay bills. I like to.
No. Go ahead.
Yeah, but anyway, so as in done, Britannia is committed, right? She's invested. I always say, mommy stays. So, like, you think about the little circle being the child. Mommy keeps the child, but daddy runs.
All right. Okay.
So way back to my CSEC days, that is what it was. And I have never forgotten it. It's ridiculous. It is sexist. It is, that is not the case. We have good, upstanding men in society and fathers and whatnot. But I mean,
daddy going for milk.
Yeah, right. Well, you see, daddy will go run all the errands, and mommy will stay and babysit. You
well, I'm sure our viewers getting that. They're not going to mix up that anymore.
I told you it was terrible, but it worked. Yeah, so we have our possibilities here. What is the chance of them having colorblind children? So I'm going to circle in a different color all your possibilities of having a colorblind child, which is going to be from 2 and 4. Right? So the chance of them having colorblind children, they didn't care about the sex of the children. So you have a 2 in 4 possibility. The chance. You have a 2 in 4 possibility of having colorblind children, or 50% chance. And then, so that's it there, 50%.
All right, let's jump into the next question. I'm supposed to have a maths live starting 12, so that's the cutoff time ish. Yeah, so we just revising inheritance alone, but it seems like we have lots of different types of inheritance questions. What's the percentage of our inheritance, the probability of an inheritance question showing up?
Probability. It's pretty good. It's not going to come in a big way. It's always a part of a question. And it's usually, it could come anywhere between like 5 to 9 marks of a question.
And how much is the total marks in the bio?
SB 50, I think 15 in a particular.
No, I mean in the entire exam.
Oh, I can't give you the answer.
Okay.
Yeah. So this one, the plant from which the flower shown in figure 1B is a part of a question, but the actual plant is not important. Exists in 2 varieties, one that produces blue flowers and one that produces white flowers. A cross between 2 blue flowered plants produces 100 seeds, and 74 of the plants that develop from these seeds have blue flowers, and 26 have white flowers. And from that information, they want you to deduce the genotypes of the parent plants. Explain your answer with the aid of a genetic diagram.
Why not 75 and 25?
So that's the thing. 75 and 25 is ideal or Mendelian, right? But the thing is, actual genetics don't usually pan out to your expected results, but it approximates. So then you usually do
okay. I don't understand.
So you do a statistical test, to do something called a chi-square test that shows that it follows.
But the larger the population, the closer to the
ideal ratio you will get. Yeah. So you can have little environmental influences, things. It will always be exact. And you will see it more, as you say, in larger populations. So based on this, you basically have from this 74 to 26 ratio is approximately your 3:1 phenotypic ratio in your F2. Right? This is where your familiarity with all the possible standard combinations when you cross two homozygous dominant parents, when you cross two heterozygous parents, what it is you're going to get, right? So in this case, when we get this 3:1 phenotypic ratio, this is telling us that, so from this, we have to know from our common crosses, if you cross two heterozygous individuals, so we're talking about blue flowers, let's say we had a cross between big B, small b by small b, right? That means the four possibilities we're going to end up with would be big B, two possibilities of big B, small b, and of small b. These three codes for your blue flowers, and this one is going to code for your white flower. Yeah.
Great.
Now you could answer this question by process of elimination, but that's just a waste of time. If you are familiar with the phenotypic ratio, you know what the genetic cross would have, that would give you that, and therefore your first line of evidence would just be the one that's going to be correct, right?
I
So
just going
in
to
this
get
case,
something on my
again,
phone.
Right. So in this case, you're going to go ahead and let your big B equal blue flowers, right? So one parent, your parents are going to be heterozygous. So this is going to be a blue parent here, right? Which is goes in line with what the question says, because it told us two blue flowered plants give you this outcome. So the Punnett square that we draw here show so this, guys, is what I know from my studying, and then I know that these are the two genotypes that I need to cross to show in my genetic diagram, right? That would give you these ratios here. So both parents must be the 74 blue. Yeah. So with that, we could move on to, I have some sample MCQs that we could run through really quickly that kind of span a lot of the different types of questions that we went through. So it will show pretty quickly if we kind of grasp the little differences in the different types of inheritance questions that can come. I'm not sure I can say which topic is worth the most attention, because with the biology exam, because they do piece of different topics that can be connected, obviously I will say that if you have to focus on a particular section, your section B would, mastering a section B would give you your most marks, but then that means that section B is the largest of the 3 sections. So I wouldn't jump out and say a particular topic, and not saying that you shouldn't study section A or section C, but section B is where most of your marks are going to pull from.
Hey,
Right?
when is bio exam?
CSEC Bio is on...
I know physics is way down in the end. I think chem kind of down in the end too. When is bio? Because they're asking if to do our next live now, and I feel like we could probably do one where we could probably do a short video, like and just kind of give them a little vibe on what to focus on, and then we could probably touch another topic. I think what topic you were suggesting?
The paper 2 is on the 22nd.
22nd.
We could change the slide, or we want to wrap.
Leah, let me jump into some multiple choice now.
Yeah. So we went through the blood groups already. So then we should be able to figure out this one, right? Tarzan and Jane are heterozygous for blood groups A and B respectively. So we know what the genotypes of Tarzan and Jane are, right? Which of the following shows all the possible results of their genetic cross? In the chat, what do you think? Are all going to be 100% big A, big B? And we talked about that possibility. Somebody asked a question about if the parents are homozygous to type A and type B. Is it going to be B, 50% A, 50% AB? And well, I carry out all them possible nuances. But what do you think?
I'm idiot because I know what the answer is. Yes, the chat going in.
Right? So I think everybody's saying these in the chat, which is what we should, right? B to B means that you can have a child on all 4 possible blood groups, equal chance. Yeah?
Next one.
So second, which of the following options correctly describes DNA, chromosome, gene, and allele? So I didn't really go through any questions like this because this is more like definition things you learn, right? But understanding these definitions would clearly allow you to understand the codings in the other questions. You know what they mean when they use these words, right? So what is which one correctly describes DNA, chromosome, gene, and allele?
Chromosome, gene, and allele. I don't think I can get this one correct, and I might have to guess, believe it or not. Just now, right? The key here describes.
Yeah. So everybody in the chat saying A. Right? But this is where I always say you have to read your question. You saying A too?
I'm not feeling A because I find deoxyribonucleic acid not really something like I described in the first place.
Yeah, that's just saying what it stands for.
I like D.
You like D. So I don't like A and I don't like D.
All right. So what is it?
Right? So the answer is going to be one person. No, Shanae and Kezia in the chat. Right? Says C. It's going to be C. A gene is the smallest unit of inheritance. One, that's a fact that we know. We know that alleles are different forms or alternate forms of the same gene. Whether it's going to be the gene is B, but whether it's big B or small b, that's different alleles. Right? With the chromosome is DNA wound around histone proteins, not A that has histamine. Right? So that's, and then DNA is the nucleic acid that has all the genetic information. Yeah, okay. So it's going to be C.
C.
You had to read your options carefully. They use histamine instead of histone. Sneaky. If a diploid organism has 2 different alleles for the same gene, it is described as being Knowing what this means is important for these questions. So we're not talking down, we're not talking, so it's either between A or B. That is going to be A, right? We met that term heterozygous all across the last section, right?
Okay.
Yeah, next slide. Two alleles of a gene are situated at, is it the same locus on a homologous pair of chromosomes at different loci on a homologous pair of chromosomes? So locus is the position of a gene on a chromosome. That is what locus is. And the plural of locus is loci. So I'm going to frame you up with that.
All right, I clearly need a refresher on what happening here. Like, okay, so how this going? It's supposed to be this to this to this to this. What going on here? The gene and the allele, the gene, the chromosome.
Right. So a gene.
DNA.
So you have.
You want blank slide
have,
to draw? Let me put a blank slide in 14.
No,
next slide. Already have it on 14.
Okay, so let's keep it simple. A homologous pair has the same gene in the same position. Has the particular gene on the same position on both of them. So if we have gene A, there will be a copy of gene A here and a copy of gene A here. This now, gene A, you can have
what are these things?
Many. So say in the alleles here are big A, homologous pair.
Homologous pair what?
Right? So a homologous pair comes from, so you know that we have 46 chromosomes in all of our cells, right? The 46 chromosomes come in 23 pairs because we get one copy from mommy and one copy from daddy for each chromosome.
So this is a chromosome.
A maternal copy from mommy and a paternal copy from daddy. So this here is one chromosome.
Okay.
So you can have big A here and small A here. And then you can have gene B that also has many alleles. That could be big B or small b. And if gene B is in this position on this chromosome, it's going to be in the same position. You know, imagine those two chromosomes exactly, they match, yeah? This exact. And they can have the same allele or different alleles. But where that allele is going to be found on the chromosome.
So this is a chromosome, and we have alleles on the chromosome, which is a certain number or some kind of thing, and then there's another
word. Genes in different positions on the chromosome.
Gene. So what's the difference between gene and allele?
So gene is okay. You have a gene that's going to govern hair color, right? Different alleles for the gene for hair color means that you can end up with black hair, can end up with blonde hair, can end up with red hair, can end up with brown hair, right? So alleles are alternate forms.
So like the allele is like the position in the house, and the gene is what it is, like what is in the house, like whether it's B or small, what you understand what I'm trying to say?
So
that is
fair. The gene is the house, right?
The gene is the house, and the allele, okay.
Yeah, so we all have the same genes.
Okay. So
we all have the same genes, but we have different forms of those genes, and that is what accounts for us being different from each other.
Okay.
Right, so if gene A is the gene for hair color, big A might give you a brown or black hair, which is most common, and small a might be, but obviously it has more these
Yeah.
characteristics have a lot of alleles, right? So you can
Right.
have a lot of differences. So Timothy's saying an allele is a different flavor of the gene, right?
Okay.
So you have ice cream, you can have chocolate ice cream, strawberry ice cream, pistachio. I mean, after chocolate, there really isn't any other, but.
So where does DNA come into all of this?
So DNA, one chromosome is one extremely long piece of DNA.
Ah.
So you have one string of DNA, that's your chromosome. And then that DNA is wound around different histones. Describe chromosome before we said DNA plus histones. The DNA is wound around these histone proteins, and then it's compacted, it will fold up, heavily folding into a chromosome.
Okay. Let me see if I have any more things to get out of my system here. Ting.
So we have 46 chromosomes in all of our cells. We have 46 pieces of DNA in all of our cells. And a gene is just a gene is just a stretch of DNA.
Okay. Okay. Wait now, is you try to pass off chocolate as a viable flavor of this life had a finish.
I feel like that moment passed, the moment passed.
Chocolate not even anyhow. Let me not this can it's all smiles right now. So let me keep it going. Next question.
Two goats heterozygous for fast growth rate are crossbred. The percentage of the F1 population which possesses homozygous alleles is
it's actually 50%. I feel it.
Yeah,
Right.
because
25% was tempting me initially there.
It would be, right? Because say we're doing this is the cross, right? You will end up with big G, small g. Homozygous dominant, and homozygous recessive. So these two will give you a 50%.
So like if you had it like this, have a GGD and a small GD. These could be the mixers.
So if you jump out with your Punnett square, that's where you end up.
Okay. Next.
Homozygous, the two possibilities where the two alleles are the same. In guinea pigs, rough coat is dominant to smooth coat. If two heterozygous rough coat guinea pigs are mated, they would most likely produce
So most likely means highest percentage.
when you watch your options.
Rough coat, offspring only. Smooth coat, offsprings only, and equal
So
more rough coat. An equal number of rough coat.
D. Yeah, so you have 75% rough coat, 25% smooth coat.
Yeah, because
so you
the
have more
dominant
rough coat offspring than. No. Yeah.
Nice.
Last. The item refers to the following diagram, which shows a section of a pair of homologous chromosomes that code for hair color. Right? Which of the following describes the genotype shown above? Easy. Well, I say easy just because it's because what we kind of just discussed it.
But when they say homologous chromosomes, they just mean the pair.
Yes.
Okay. And this
but when they say homologous chromosomes, they mean that these two chromosomes have the same gene. Sorry, they have genes in the same position, but they may have different alleles at that particular gene position or gene locus.
So it is big. Well, that feeling heterozygous to me.
Whoop, you see a difference. So once you have a difference, it is heterozygous. That's B.
What would homozygous dominant be? That's a thing? Oh no, homozygous
Yeah,
dominant is
homozygous
AA.
dominant
Okay. This is
is
hetero
AA,
homozygous.
and small a would be homozygous recessive.
Okay. All right, we could keep pumping. 15.
Hemophilia is a sex-linked trait caused by a recessive allele. Which of the following are reasons why the disease is expressed in more men than women? So now you see if you're pulling.
I'm feeling like I getting one-on-one tuition here, boy. Like I'd pay money after this. And it's caused by a recessive allele carried on the X chromosome. There is only one X chromosome in males. Women have two X chromosomes. So wait now, I find all of these sounding like contribution to the reason. Yeah, I get them the 1, 2, 3.
I'm hit them the 1, 2, 3 as well. Right. Albinism is caused by a single recessive allele. Two normal parents have an albino child. This is because
both parents were heterozygous for the gene. Both parents were homozygous recessive. That's impossible. Otherwise, they'd have been showing it. One parent was homozygous dominant for the trait. No, otherwise he would have the masking thing would have happened. Oh, one parent was homozygous. No, so it has to be A that there was some heterozygous vibes going on here. So that's 25% albino. Yeah.
A girl has blood group A and her brother has blood group B. Her mother is blood group AB. They both have the same father. Which of the following are possible genotypes?
Oh, I thought they said the mother and the daughter and the same father. It's the mother and the brother. Okay. A girl has blood group A and her brother has blood group B. Right. Good. Her mother is AB. Okay. Okay. All right. That clears up that. So if mom is AB and the brother is B and her and she is it has some, I feel like it has some going through this question, but like I need to think about it. Just know, miss. So AB, if we have A, well then we'll have a and a B. No, you can't have A. Because if I have AA, this is what I'm visualizing. I'll get some dominant homozygous here, and then I'll get some co-dominant kind of. What is called that when it's both of them dominant? What is this? What is AB? What do you refer to that as?
AB is a different blood group, right?
It's a whole different blood
remember,
group.
You have to be able. So this possibility, there's no way to have a boy, a group B boy.
The BB, same thing will happen in reverse. So let me try the AB now. AA. So wait now. A, B. No, what am I trying? AB, yeah, BB. All right, so this one could work and this one could work. 3 and 4. And I know it had to be 3 and 4 because if 3 working, that, well, 2 now working. Wait now, you could kind of reduce because since double A now working, double B can't work. So you kind of get the other 2 by just watching the answers.
You end up with 3 and 4.
Yeah.
Yeah?
So you're getting the situation here. This is brother, this is sister. And with O ain't saying nothing, so AO, that is the sister.
There are 2 possibilities there.
And that the brother. Nice. We have any more?
I think about 4.
All right, let's jump in. Let's do this. Slide 16.
So this item refers to the following diagram, which represents a cross between two plant heterozygotes for a particular characteristic, and they're asking you which of the following completes the diagram at 1, 2, and 3.
Oh, I don't understand the question. I thought for a second that one was like a thing. I'm not going to ask you what is that.
What we're not here.
Yeah, but that's where they want to put the answers. So they want you to put the answers there, right? And across.
So because they tell us the two parents are heterozygous, one has to be
big R.
Right.
So I could eliminate A and C.
So big R has to come here. And then 3 would be when big R and small r comes together. 2 would be small r and small r. So your answer there.
Maybe. You know, I love to do multiple choice after I learn a topic.
The gene for code color.
After I do the paper twos, like coming off with the multiple choices, feel like okay, you put down the heavy work and now the multiple choices adding in the final touches is kind of bring everything together.
Right. But I did that this way as well because a lot of students sometimes, when some exams, very few, but in some exams, most of it happened with the CAPE students more than the CSEC students, but their paper 1 is before the paper 2. And if you didn't do all that mental work when we did the paper 2 questions, imagine just trying to jump out and answer your paper 1 questions with, like you see in this paper 1, so you didn't have to study as hard, you didn't have to go into, you don't have to study as deep, right? But if you didn't do all that brain work before, all them tables and things that you kind of doing mentally now, you'd have had to draw out in a paper 1 exam for that 1 mark. Yeah. So paper 1, you have to take it as seriously as your paper 2. Yeah. The gene for coat color in cattle shows incomplete dominance. A purebred cow with red coat mates with a purebred bull with white coat. All of the offspring have rowan coat. So rowan is a mix of the red and the white. Which of the following would represent the genotype of the offspring? So this question brings in a notion of something that we didn't meet before, which is incomplete dominance. So we've met before where you have a dominant allele and a recessive allele, and once the dominant allele is there, the recessive allele isn't able to have influence or show its characteristic, right? Incomplete dominance is where one allele is slightly
doing a thing,
have a stronger effect, so they both battle. It's not complete dominance where one masks the other, but the phenotype that's going to result from it is a blend,
a mix.
So Jaden is saying incomplete dominance and codominance are different, right? Because Timothy was saying, I am assuming, he gave an answer, I'm assuming incomplete dominance is the same as codominance, and that is not true. Like, codominance is the fact that you have blood type AB because allele A and allele B have equal strength. So they both show,
effectively they create a whole different blood type for that. AB.
Right. Incomplete dominance also does create a different phenotypic class, but in incomplete dominance is supposed to be a blend. Right? And for some reason, at the CSEC level and at the CAPE level, whoever is setting the questions, I feel like they don't understand fully incomplete dominance and codominance.
Okay, right? Get into the question a little bit, how they phrase it.
Because incomplete dominance means that you see a blend of the phenotype. You don't see either one, but it's a blend. So, for example, if one parent is red and the other is white, in terms of flower color, incomplete dominance should give you a pink flower, a blend of the two.
Okay.
Codominance means that you see both of the parental phenotypes. You see red and white. So codominance will give you a red and white flower, and incomplete dominance will give you a pink flower. The example with the cow and the bull here, giving you a rowan, is where you have patches of red and patches of white, where you're seeing the two, which is codominance. So
okay.
The question is wrong. The genetic basis of the question is wrong. However, the genotype is the same for the both of them. It's just how the genotype is translated to the phenotype is different. So you need a heterozygous. So the answer is B, but they have
divide
a
by
error in the
the
question.
they're off.
Yeah?
Yeah.
Zane, the son of James and Jennifer, may have inherited the red-green color blindness gene from his parents. Neither James nor Jennifer is colorblind. If N represents normal, what are the genotypes of the parents?
That start off like ecclesi King
It's
Suit
called
and
biblical.
neither James nor Jennifer is colorblind. So both of them not colorblind, but they have inherited the red green color blindness, but they're not colorblind. So so what are the genotypes of the parent? I would think that that pups had to have a capital N, because I find when pups have the common N, son in problems right through. So he had to at least have this. So the mom is the one who have inherited the red green color blindness gene from his parents. The sons of James and Jennifer may have. Zane, the son of James and Jennifer. I misunderstand this question. Neither James nor Jennifer is colorblind. So may have mean that Jenny is the one.
Just remember, James and Jennifer are both, they are the parents. So,
Okay, neither James nor Jennifer is colorblind. So James, if he not colorblind, he must be this, right? So it's either this or this. And if the same may have inherited. Yeah, so and Jenny gotta have the big end too. So I feel an A. How A song then?
A something like it for me too.
Mm.
Right? I think the last slide that I did have was to touch on mitosis.
Oh, so the last one. Okay.
Yeah, it's just to touch on mitosis to bring the different stages to here, but this could really balloon into a whole different topic. Right? So I mean,
what's the answer for this though? The correct order of this.
Again, with mitosis, the acronym that you really want to remember is, most teachers teach it. You have to remember your.
No, look at me. I figured a long time. I know it had an acronym, but I figured that. You can find it on my YouTube videos.
I think my thing's sticking in. Allow me to write, but you'll remember PMAT.
Right, yeah, PMAT.
Right? So PMAT stands for prophase, metaphase, anaphase, and telophase. Right? So that's the order. Right? So again, you see, Arissa put a comment in the chat called IP. She put IPMAT, right? The I, that interphase, is not a stage in mitosis. Interphase is what the cell is in between mitotic events. Right? So if you have to give the stages of mitosis specifically, you have to talk about prophase, metaphase, anaphase, and telophase. Prophase, certain things happen. In prophase, your chromosomes shorten and thicken, they become visible, and that nuclear membrane, I can't draw, right? Because
you can see again.
Pardon?
Oh, that we're going to a whole thing. But check and see if you can write on the slide again.
Nah, I see in the circle dancing by me here.
Oh, okay. I might have to leave and come back in that part.
RL out.
Okay.
But 3 is going to be the first one. 3 is showing you prophase, where you're still seeing that nuclear membrane. So you're seeing your 2 chromosomes there, they're visible, but they have the circle around it. That's the nuclear membrane. So if you're still seeing the nuclear membrane, that means you're in prophase. During prophase, that nuclear membrane breaks down, right? Then, after that, your chromosomes are going to line up on the equator of the cell. So you see in 2, where they kind of one on top of the other there.
Mm-hmm.
Right? And then after metaphase, now you go to anaphase, where they started the chromosome splits and the chromatid goes, one chromatid goes to one side, the other chromatid goes to the other side. So you add 1. And then finally, once you have the 2 groups on at either poles, nuclear membranes reform around the 2 sets and leave you with your telophase there. Yeah.
Vibes.
And now
Yeah,
back in
when you talking serial activating on your thing, boy.
Yeah,
That's
Sure,
serious.
I'm minding my business. Yeah.
All right, Akili. It was nice. We had to do some more. So I'll see how our best
Yeah,
our
man.
time
We could get
overlap,
something again.
and maybe we could jump in with some
Just
of the other topics.
then let me know. Let them send you topics and I can prepare a different set.
All right, sound good. Well, you're frozen, but we out, people. Love and blessings. I'm gonna jump back in probably in the next 15 minutes or less with the final stage of this maths. Remember, we have the maths live. What we're gonna do, we're gonna be doing some vectors and matrices. Love and blessings.
All right. Bye.